Thank you so much, Darrell. Thank the Congressional offices that have sponsored and supported this briefing, Everylife Foundation, and those of you who are affected by this topic. And again, it's a pleasure to share this panel with this august group of scientists and perhaps most importantly, the patients who are at the heart of what we do and why we do it. Indeed, Darrell mentioned we're the third largest institute. That's something Congress could change. We could get a little bit bigger. But in all seriousness, we do a mission with our $3.5 billion that doesn't stay here in Washington. And indeed, our charge, our mission is to turn discovery science into the health of the nation. And we do that really by sending those dollars out to brilliant scientists like you've already heard from today. And indeed, it's that investment in literally districts and states all over this country that we believe does have an incredible return on investment. The National Heart, Lung, and Blood Institute is actually also one of the older institutes. We just celebrated our 70th anniversary last year. And the genesis was to address a very common public health menace in heart disease in the middle of the last century. And as a result of the investments in biomedical research, we've witnessed a 70% decline in coronary heart disease events over that same sort of period. And that's a common disorder. Obviously, we're gathered here related to rare diseases. And I know there's often a sense that these are neglected. We use terms like orphan as if there's something that's secondary. And yet I hope what we'll also convey is that this is an important priority. As Dr. Nelson alluded to, it may be a rare condition, but when it affects your family, it's an important priority. And indeed, collectively, rare diseases are all throughout the NHLBI portfolio of heart, lung, blood, and sleep disorders. And collectively, this affects tens of millions of Americans. And so, indeed, it's a critical part of our stewardship as a federal agency to ensure that we're driving science that has an impact, regardless of whether it is, quote, a common disorder or a rare disorder. It's still fulfilling our mission. And critical to fulfilling that mission, we need that whole circle of partners that is shown on this slide. Particularly in the cases of rare disorders, the patients are at the center. And I'm so pleased that Tisha Samuels is here, because this is our prime motivation. Dr. Nelson eloquently stated how this can be a driver of discovery. It all begins and ends with the patients. That's why we do what we do. And indeed, we need the patient interest groups, the advocacy groups that are represented here, is the citizens. I consider you collaborative citizen scientists with us to help drive this agenda, because again, that's critical to us fulfilling our mission. You've also heard that we need really a collaborative ecosystem of partners, both in academia, in foundations, organizations like yours, the private sector, all working together to really try to solve these problems. So very briefly, I'll outline a few exemplars that come from our portfolio. First, starting with some of the legacy of what we think has been a successful period of time over our 70 years that highlight two exemplars of rare disorders, one cystic fibrosis shown on the left, the other sickle cell disease, where often when I was a Harvard medical student, these were almost death sentences in which it was thought that it'd be very difficult for a child born with these disorders to live into an adulthood. And yet, over the course of my career, we witnessed tremendous lengthening of the life expectancy for both cystic fibrosis and sickle cell disease over this time period. And again, it's reflective of that virtuous cycle of investments in fundamental biomedical research that have identified pathways and processes that promote the disease, and then developing smarter and more targeted therapies that then are important in ameliorating that whole course. And both these disorders have seen that play out over the last quarter century or so. And so as we build upon that sort of legacy of success and excellence in biomedical research investments, we're looking forward to the horizon that we find is very exciting, very promising. And you're already getting glimmers of it here that really bode well, I believe, for a variety of rare disorders, of which we can only touch on a few quick examples today. And it's part of the strategic vision that we have for the NHLBI over the next 5 to 10 years. And this was a strategic vision process that was very inclusive, in which, indeed, citizens, patient advocacy groups, policymakers, professional organizations, all were brought together to identify what are the most compelling questions and critical challenges we need to take on in the next 5 to 10 years. And how can we bring to bear emerging new technologies that could be real game changers, that could be transformative in the lives of patients with both common and rare disorders. And indeed, it's innovators like Dr. Church who are bringing to bear those new technologies to make questions that we couldn't even address a decade ago really proximal and feasible to take on now. And that's what we're pursuing. I'll give you just 3 examples across our heart, lung, blood, sleep spectrum in each of those domains to give you a glimpse of how we're leveraging these new technologies and tools. One of those examples is shown here, in which, as Dr. Nelson alluded to, Duchenne muscular dystrophies unfortunately follow a life course where a key muscle is obviously the respiratory muscles, the diaphragm, and so lung failure can be an important and unfortunately cause of demise eventually. Also can affect the heart and indeed promote a cardiomyopathy. So as the director of NHLBI, 2 of the major morbid and death-causing complications of Duchenne comes back to our portfolio. And indeed, we are encouraged by advances in what we're making in terms of understanding, again, what the cause is, how that protein works normally in normal biology, how indeed mutations can affect that protein, and how that dysfunction affects the cell that it's in, and how that may then allow us to see how can we take this as a therapeutic opportunity to see how we might normalize that cell's function again and reduce the sequelae of the symptoms and perhaps prolong life. And so, as he's alluded to, we're very excited now about the technologies that Dr. Church alluded to, so that now that we can see that misspelling of the genetic code that might predispose to a rare disorder, the technology now exists to almost like a word processor to go in and if there's a misspelling, to correct that spelling. And again, the tools are now available, CRISPR that you've heard described, that's basically like a molecular scissors that can precisely go in to where that misspelling is, cut out the misspelled letters and words, and then provide a way of replacing it with the more normal spelling of the genetic code so that normal protein is generated and normal function is restored in ways that hopefully will restore normal health and viability. That capability, again, was hard to discern as possible really 15 years ago. And indeed, I may make as a side note, it's intriguing that that technology was not necessarily discovered because we were trying to solve sickle cell disease. It was discovered because someone was studying how bacteria kind of defend themselves from invading entities that had their own nucleic acids and DNA. And they figured out that a bacteria could protect themselves by using these scissors against their enemies that are invading them. And so then it took basic scientists studying bacteria's defense systems, to then say, hey, maybe this could be used in a therapeutic way. And so I highlight that because that research was not related to a particular disease at that time. It was fundamental discovery science that allowed us to appreciate how these molecular scissors work in the first place. So it's always critical, as much as we advocate for a particular disease to be cured in legislation, it's also important that we also never forget the importance of funding fundamental discovery science that allows us to just understand how basic biology works, because ultimately that's going to be the key to finding these cures. I would also indicate that this is a strategy that continues as a recurring theme here, in which, again, cystic fibrosis has followed the same virtuous cycle, in which, again, a debilitating condition, in which, again, the gene was identified and its important role in maintaining the conductance of ions and water across membranes was understood at a very fundamental level. And indeed, connecting that to the pathobiology of the disease, in which that compromised how these patients were able to mobilize the normal mucus and flow that's important for normal lung function. And recognizing, again, what happens when that transporter is perturbed. And indeed, understanding the biology about how different mutations affected that transporter, even how it's mobilized and moved within a lung cell. That insight into how different mutations behave, not only clinically to the patient, but biologically, set the stage for new small molecules to be developed that then influenced how that even perturbed transporter might be rehabilitated, if you will, so that it gained more function, despite never changing the mutation itself. And indeed, that's given birth to another whole new generation of therapeutic molecules that are transforming the lives of patients with cystic fibrosis. Also part of this virtuous cycle was not only that basic discovery, that understanding of pathobiology, that identification of targets, the public-private partnership, patients were critical to making that happen. Indeed, it's one of the exemplars of citizen science where, again, the Cystic Fibrosis Foundation clearly played a critical role in organizing the whole ecosystem, in helping enroll patients and being a real partner in driving the success of that science. So that example is what gives us hope, and hopefully you're hearing a sense of excitement about sickle cell disease, which is my final example related to a blood disorder that actually is relatively common amongst rare diseases. It affects about 100,000 African individuals in this country, predominantly African American, though it affects every race, ethnicity around the world. Tends to be driven by areas where malaria is endemic, because if you have just one copy of that mutation, it actually can be protective against the malaria and indeed promote survival. But then two copies really compromise hemoglobin, the protein that carries oxygen around to your entire body. It's critical for tissue function. And that unfortunately affects not only 100,000 in this country, but literally millions around the world. There are hundreds of thousands of babies, predominantly in sub-Saharan Africa, born with sickle cell disease every year. So indeed, this is a disease that I'm sure Tisha will share with you what it means to live with this disorder. But again, one that affects so many lives and families. But again, we're encouraged that by it being actually defined as one of the first molecular disorders back 1948 or so. And we've known a lot about its genesis. But it's taken us a while to really develop a precision medicine sort of therapeutic strategy that could really transform the disorder. Indeed, we've been committed to really a full court press on this disease for some time, but again are encouraged by the emergence of new technologies that are now clearly here on the horizon. There have been several strategies. One of those strategies relates to what we've learned about the natural history. The clinicians have known for some time that the course of sickle cell disease could be influenced by how much of fetal hemoglobin that a patient might have. Ordinarily, you have that fetal form just when you're in utero, in your mom's womb, because that's great at dealing with the oxygen condition in that context. And that gets shut off, and you turn on your normal hemoglobin adult form for the rest of your life. What's been learned is that presence of that fetal hemoglobin seems to protect against sickle cell disease. And that was actually a clinical clue about what might be an effective therapeutic strategy. And so, as shown here, it's starting to emerge that we might be able to use these new tools and technologies to take a couple of different strategies. One may be, can we genetically engineer the normal bone marrow cells that produce these red blood cells to have a boosted amount of this hemoglobin F, this fetal form, that might prevent sickling and indeed have a more benign course, if you will. So that's actually a strategy, again, using these CRISPR technologies that is now currently being tested. And indeed, there's actually another genetic or heritable entity called hereditary persistent hemoglobin F. So it's almost an experiment of nature that we know that a precise place to put this mutation. And again, it relates to some basic biology. It's actually elegant that Dirksen has done in terms of understanding what are the ways that these genes are regulated. So now we can go in and actually turn on and turn off and manipulate that gene's expression in a way that can be beneficial. And so we're very excited about that. In addition, other strategies to actually fix the mutation or replace the gene. So what I've tried to convey to you with this last example is an initiative we're pursuing that involves a public-private partnership, a lot of collaborative opportunities with the FDA, in which we're committed to curing sickle cell within the next 5 to 10 years and pursuing clinical trials to bring that about that involve both the public and the private sector. And we're very excited that Keisha's here because she's an exemplar of some of those strategies that are being pursued. In many ways, this is a forerunner of what we hope can be part of that virtuous cycle that can be extended to a variety of rare diseases. And all of that will come about as we continue this work with our circle of partners, policymakers, patients, the private sector, and your organizations to make this transformative moment come sooner than later. So thank you very much for your attention.